There should be no reason for you to leave any question empty. If you're not already familiar with the multiple choice answer sheet, you should be. Okay, I will send this. See if I can send it. And this sheet, you have to get used to it for the final exam. Yes, the final exam will look a little bit different, but same spacing, generally speaking. And you should factor shading time into your overall solve time. Let's see if I this. The sheet to the chat here and to the ASA level bio and to this chat. You should be familiar with this and you shall print it. Can you guys hear me or am I still lagging? Okay, so there's the multiple choice answer sheet. I also sent it to the ASA level biology chat. You should print it and be familiar with it while solving any paper one exam. And the key in paper one is to solve as much as you can and not just mark, oh, I got it right, I got it wrong, end of discussion. No, you should mark and find out where you went wrong, why your choice was incorrect, and why the correct choice is correct. Now, let's go to a few guidelines. Now, these are just suggestions. They are little guides to help you throughout the paper, and you don't have to follow them, but this is just my advice. First off, when reading the question, you want to read the context first, and the options, and then the question, and then the options. What do I mean by that? Let me get any question. Okay. Question 5, for example. This is the context. These are the options. And the line with the question mark, that's the question. And again, these are the options. So your eyes are going to read this first, and then this. And then you read the question with the context of the options. And then you go back to the options. That way, you get the full scope of the question. You get all the context necessary while reading the question and while rereading the options. Second thing is do not work on the process that all the options are wrong. I'm trying to find the one that's correct. No. Innocent until proven guilty. You work by process of elimination. We as humans are more prone to finding the incorrect, to finding the little scrutinies than we are at finding what's correct. So that's why we work by elimination, not by possibility. We find what's wrong, and then what's left is the correct answer. And many questions here will use that scheme of work. You may not understand all the options, but you may understand enough to eliminate a few, and you'll be left with one, maybe two. And we'll discuss that edge case later. And this, I cannot stress it enough. Even if you're 100% sure the answer is A, you will still read B, C, and D. As I said, you have sufficient amount of time. You used to solve the same number of questions in 45 minutes. Now you have almost double that. So you will read all the options. Make sure to cover all bases. Who knows? Maybe you misread something in A and the answer is actually D, etc., etc. Fourth thing is don't be afraid to annotate on the question paper. Sometimes we like our question papers to be completely white, completely nice with just the circles where the answer is filled in. This is the question paper, not the answer sheet. The question paper, this one, not the answer sheet. So you will annotate on the question paper. That's totally fine. Don't be afraid to do it. It'll be much better, and you'll see me annotate while solving. Okay? The answer sheet is the only thing where you're only allowed to shade. No comments, no annotations, nothing. You will just shade in the box. Like so. And that's it. For example, if the answer for one was A. The last key scheme I want to emphasize, this is really important, is don't change your answer unless you're 100% sure it's right. If I set first answer to A, and then while revising, oh, I feel like it's B, don't change it unless you're sure A is wrong and you have a reason for why A is wrong. Otherwise, keep it as it is. It's preferable not to use highlighters in the question paper because it may cause problems. Just use use pencils. Okay. We're going to start the paper. We're going to pass through all the options. For calculation questions, you don't work by process of elimination. You calculate. It's not any weird thing that you're going to follow. So the eyepiece particule for a light microscope times 100 magnification. You have a set magnification, eyes on it. Let's see if it changes because that'll change the answer. Okay, was calibrated using a stage micrometer. Each small division was 0.01 millimeters. Now this removes the idea that every micrometer scale, the small division will be 0.1. Here we have an example, it's 0.01. Now, while working here, we always work by cross multiplication. How many eyepiece graticule units are to how many millimeters or micrometers? So let's first start. We know that each small division, so every one of these is 0.01 millimeters. So I'm going to simply get a ruler and draw a straight line here. And here, and oh, I see that it doesn't line up perfectly. It's spaced. I don't use it. I find some place where it lines up perfectly, as close to perfectly as possible. Here we have 30 eyepiece graticule units. 2, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 divisions. 10 times 0.01 would be 0.1. So 30 eyepiece graticule units is 0.1 millimeters. Single cell organism was viewed with the light microscope at the same magnification. Thank God. There is no there are no plays with the magnification going on, so we're very hopeful here. The length was measured 47 units. So we're going right here, 47 EGU would be how many millimeters? And then normal cross multiplication. 47 times 0.1 over 30. That would be 0.156, roughly 0.16 millimeters. Okay, now I look at the option. I can't find anything. I find 1.6A. Look at the units. Micrometer. That times 10 to the -1, so that would be 0.16A. Look at the units. Here it's micrometer, here it's millimeters. So we work accordingly. And we convert from millimeters to micrometers. You know that this conversion is times 1000. 1, 2, 3. Decimal point 3 places would be 1600 micrometers. I can't find a 16. Sorry, that would be 160. 160 micrometers. I can't find a 160 here. Then we work by converting the standard form to normal digits. Times 10 to the negative 1. Obviously, this is 2.9, so it's not even into our consideration. Here, times 10 to the negative 1, that would be 0.16 micrometers. Doesn't work. Here, times 2, 1, 2, 0 here. That would be 160 micrometers. The answer is C. Yeah, magnification times 100. Also, while calibration. If the magnification changes, then we have discussions to go. I hope someone sends the question. But if the magnification is the same, then we calibrate. Then it's the same calibration and there's no change. Now, one of the electron micrographs shows a eukaryotic cell, the other shows a prokaryotic cell, and this is the context. The image is part of the context. Okay, which one here is prokaryotic and which one here is eukaryotic? Now, obviously, the one with the nucleus is the eukaryotic cell. And we see here a very thin cell wall that doesn't signify anything. Okay, but there are no nuclei here, and there's a huge nucleus here. So we know that this is going to be the eukaryotic cell, and this is the prokaryotic cell. What is the approximate distance across the eukaryotic cell at its widest point? This is another calculation question. You're going to get your ruler. And the widest point would be from here to here. That's about 80 millimeters. So that's 83 millimeters. Now we know that actual equals image over magnification here. So 83 millimeters. I'll write it down, sorry. 0.02 7 0.05 1,000 1 2 3. That would be 20.75 micrometers, or roughly 21 micrometers. D. Moving on, which polymer is a major component of the cell walls of typical prokaryotes? That's peptidic glycan or murine cell wall. Both names are accepted. You will find peptidic glycan more. Cellulose, that's for plants. Glycogen, it's nothing. Suberin is the waxy band that forms the Casparian strip in the endodermis. Which features are shown by prokaryotic cells and also by eukaryotic cells? Anything that's shown by prokaryotic means it is non-membrane bound. Cell surface membrane, obviously, all cells have a cell surface membrane. It what separates the cell from the environment around, otherwise it's just a pile of mush. Okay, so any option that does not have one is automatically wrong. D. Lysosomes. Now someone may say, oh, lysosomes, of course. And I'm going to say, absolutely not. Lysosomes are membrane-bound organelles. They have a single membrane with the hydrolytic enzyme N is automatically wrong, and that eliminates A. You have a cell membrane and 70 S ribosomes. They're obviously present in prokaryotic cells. They're the only ribosomes that are present in prokaryotic cells. But are they also present in eukaryotic cells? Yes, they are. In mitochondria, and chloroplasts. Therefore, the answer is 1, 3, and 4, B. Proteins. Hello? Am I lagging now, or is everything all right? Oh. How about is this lagging? There are two places for protein modification. I am taking. Okay. Hello? Can you guys hear me? Okay, 1 second, please. Okay. How about now? Am I still lagging? Yay. Okay. Moving on. There are two places where proteins are modified in eukaryotic cells: the rough endoplasmic reticulum and the Golgi apparatus. Now, the rough endoplasmic reticulum is the initial post-translational modification. It's what forms the secondary structure, among other things, of course. So, which cell structure is responsible for modifying proteins before they're secreted? Plasmodesmata is simply a thin strand of cytoplasm between two cells. So this is a plasmodesmata, and it allows simple diffusion to another cell. So no, it doesn't modify proteins. Tonoplast is simply the membrane of the vacuole. Obviously not. Golgi body, yes. Lysosomes, no. Lysosomes contain proteins, which are hydrolytic enzymes, but they are not. But they are not in and of themselves responsible. So the answer is which row shows the types of membrane that surround the mitochondria and the nucleus. There are three double-membraned organelles in your syllabus. Number 1, the nucleus. And the membrane is called the nuclear envelope. Number 2, chloroplasts. And number 3, the mitochondria. Now, an interesting premise is why the chloroplasts and mitochondria have a double membrane. Now, we know that they were originally bacteria, according to the endosymbiotic theory. So, when they got endocytosed, when they got endocytosed by the original cells, okay, the membrane of the endocytic vesicle remains present. Remained present. So this endocytic vesicle membrane, in addition to the original cell surface membrane of the bacteria, of the original archaea and cyanobacteria, these are the ones. This membrane forms double membrane of the mitochondria, and the nucleus has a double membrane for protection. So mitochondria double and nucleus double. The answer is B. Moving on. It is suggested that primitive prokaryotic cells may be ancestors of certain organelles in eukaryotic cells. Now, this is the endosymbiotic theory that we just discussed. Which organelle is most similar to a typical prokaryote? We just said that they are the chloroplasts and the mitochondria. So if we find either of those, that's the answer. Golgi body, no. Lysosome, no. Mitochondria, yes. Nucleolus, no. Which of the sugars, fructose, glucose, and maltose, are reducing sugars? In your syllabus, all monosaccharides and disaccharides are reducing sugars except for one, which is sucrose. And this is because the active group of sucrose forms the glycosidic bond, so it's no longer active, so it's no longer a reducing sugar. Fructose and glucose are monosaccharides, so obviously they are reducing, and maltose has the active group still free, so it's also reducing sugar. The answer is A, all three options. 1 second. Sorry about that. Moving on. Which molecule in a muscle cell is rapidly hydrolyzed to produce glucose? Can you guys still hear me? Hello? Perfect. Okay. So which molecule in muscle cell is rapidly hydrolyzed to produce glucose? So we have a few things to note here. Muscle cell, meaning it's an animal, not a plant. And the store of glucose in an animal is glycogen. We have our answer. Amylopectin? No, that's in plants. These two are starch. Cellulose, that's cell walls, also in plants. The answer is D, glycogen. Diagram shows how the beta-glucose monomers of cellulose are linked to each other. We see the alternating pattern. One time up, one time down, one time up, one time down. Each alternate glucose each alternate glucose monomer is rotated 180 degrees relative to the one next to it. Now, the OH groups on carbon 2, this is carbon 1, and this is carbon 2. So the OH groups on carbon 2 are on opposite sides of the cellulose molecule. One down, one up, one down, one up. What is the main way in which this arrangement contributes to the strength of structures made by cellulose? The fact that I have OH groups here means when I have another cellulose molecule below it and another one above it, I can form hydrogen bonds with both to form a cellulose fiber. So the OH groups on both sides can form crosslinks within the same cellulose molecule? Nope, with other cellulose molecules. With different cellulose molecules, through the formation of glycosidic bonds, absolutely not, they're hydrogen bonds. The OH groups on both sides of the cellulose molecule can form crosslinks within the same cellulose molecule. Same cellulose molecule? Obviously not. We said different cellulose molecules. And the OH group on each side of cellulose molecules can form crosslinks with different cellulose molecules. Yes. Through the formation of hydrogen bonds? Yes. One of the key things when solving a question is identifying the different elements in each question. Here we have the only difference are same cellulose molecule and glycosidic bonds. So this could be a table, literally. I need a blank page. Okay. This could literally be a table saying type of bond and on which cellulose molecule. Hydrogen, glycosidic, hydrogen, glycosidic, same, different, different. And this triglyceride molecule to their function. That is correct. We know that one moment until the light goes away. Is it any better now? Yay, okay. So they function as energy storage because they have a large number of C1H bonds in their fatty acid tails. Now, when you guys take A2 biology, or those who are giving composite this year, know that because they have more CH bonds, they are more reduced, so they can reduce more NAD. The key is the CH bond has a lot of energy, so upon breaking it, a lot of energy is released. Just a quick overview. Very vague. Okay, so the answer is A. We're going to still read the other options. They can function as energy stores because large amounts of energy are required to hydrolyze the ester bonds. No. And if there are energy stores, do we need energy to break them down, or does hydrolyzing them or breaking them down release energy? Releases energy. Oh, 1 second. Hello. Sorry about that. Am I still speaking? Can you guys hear me? Okay, perfect. So if something's an energy store, Does it release energy, or do we need energy to break it down? Releases energy. So B is just nonsensical. They can provide energy to all the cells because they are non-polar molecules that dissolve in water. That doesn't make sense. How can a non-polar molecule dissolve in water? This is wrong. Triglyceride molecules can provide energy because they are polar molecules that easily diffuse through the phospholipid bilayer. That's also not true. Yeah. You have the thing and its opposition. So C and D are just nonsensical. So the answer is A. Moving on. Membrane fluidity is affected by temperature and the percentage of fatty acid tails, which combination would result in the greatest fluidity. Obviously, higher temperature. We can immediately eliminate A and C. What about the saturated fatty acid tails? Now, saturated fatty acid tails means it is a straight line. So I can pack more of these in the same amount of space. So that means it's less fluid. But when introducing a double bond, when introducing the unsaturated fatty acids, space is formed. These are called kinks. So I cannot fit the same number in the same amount of space. See here? Just graphically, saturated means no double bonds, so no kinks. So yeah, a straight line in brackets because it's actually just like, a zigzag line, bond angles and whatnot. But with kinks, it's much more spread out, much more space between the fatty acid tails, so greater fluidity. So we want less saturated and more unsaturated. Less saturated, more unsaturated. The answer is B. Moving on. Solubility in water of different regions of the cell surface membrane. Immediately read the word cell surface membrane. You draw a small model diagram. Very small. There we go. Changes with the distance of each region from the center of the membrane, meaning right here. Right in the midst of all the fatty acid tails in the hydrophobic core. What is the relationship between the distance and the solubility in water? Solubility being hydrophilic regions. Now, one of the things we can immediately eliminate is P and D, because what is the difference between here and here? They're both parts of the fatty acid chain. They're both hydrophobic. So immediately, if there's any gradient, C and D are wrong. Now, is it A or B? B, we have that the center is the most soluble. Is the center the hydrophobic core the most soluble? Definitely not. It's the least soluble. And rather, the phosphate heads are the soluble ones because they're hydrophilic. The answer is A. This question is very repeated and very common. Which row about the primary structure, secondary, and quaternary? Note this is quaternary, not tertiary. Someone goes, primary, secondary, of course it's tertiary, and they don't even look at the header. We read the entire question fully. Does not matter what our assumptions or our prepositions. One of the other important things to note about paper 1 is that you do not bring your own assumptions to the paper. If it does not state whatever it is explicitly, then you pretend it's not there. So the primary structure is literally just the sequence of amino acids, the singular chain. So the sequence of amino acids coded by a DNA molecule. Yes, the sequence of amino acids determined by the codons on an mRNA molecule. Yes. The result of translation. Yes. We know that the rough endoplasmic reticulum takes the primary structure and then folds it into the secondary structure. And then the number of amino acids? No. So we immediately eliminated D, so I'm not even going to look at these options. I'm going to read A, B, and C now. Secondary structure, the result of attraction between hydrogen atoms associated with the peptide bonds. That does not mean that they are in the peptide bond itself. We know that an amino acid will form a peptide bond like so, right? And we know that the hydrogen bonds are between this double bond O and this bond H, and they are associated with the peptide bond. But they are not in the peptide bond. So this is correct. And obviously, the secondary structure is the beta pleated sheet or the alpha helix. So this is correct. The formed by hydrogen bonding, yes. And so you're going to immediately assume, oh, it's correct. No, you read the question fully. Between R groups, absolutely not. R groups only begin when groups only begin in the tertiary, and quaternary structure is more than one polypeptide. So at least, okay, by the linking together of more than one polypeptide, sure. Always contains at least two different types of polypeptide? Not necessarily. They can be the same type, two identical copies of the peptide, of the polypeptide. So they're not different. There are two polypeptides, possibly. At least two polypeptides, but not necessarily different. This is incorrect, and so our answer is A. 15. Now, this question is really important. Anyone who left and wants to come back, please come back now. This is not going to reach them anyway, but some RNA molecules called ribozymes can catalyze reactions in a similar way to enzymes. Many of these ribozymes have other molecules as their substrates and catalyze reactions that break specific phosphodiester bonds. That's hydrolysis. Another important group of ribozymes form the functional part of ribosomes. Now, the functional part of ribosomes are the ones that form peptide bonds. So that's condensation. And they catalyze in a similar way to enzymes. Which statements are correct? Binding of the substrate to the active site depends on the complementary pairing of bases for ribozymes. This is correct. It's very similar to the tRNA anticodons and the mRNA codons. And on complementary pairing of R groups for enzymes. Now, someone may say, oh, there are complementary R group pairings in proteases with the substrate protein and the enzyme proteins in the active site. Absolutely not. This is wrong because we're bringing our own presuppositions, our own assumptions to the question. What part of this entire paragraph referenced the idea of proteases? This is a question to you guys. Was there any reference to this enzyme, to the enzyme in this question? Refers to proteases. No, there are no references in the question to proteases in general. So this is wrong because it brings our own assumptions into the question. The assumption that there is a protease. Ribozymes and enzymes can catalyze hydrolysis and condensation. We just saw the ribozymes catalyzing hydrolysis and condensation. So of course, this is correct. For molecules made up of the same number of monomers, the number of possible types of enzyme is greater than the number of possible types of ribozyme. Because there is no statement that specifies that the enzymes in question are proteases. There are no R groups in anything other than amino acid residues of proteins or amino acids. Do lipids have R groups? They don't. Do carbohydrates have R groups? They don't. So there is no pairing of R groups for all enzymes. This is when we bring our own assumptions to the question. That's why one of the first things I said is don't bring your own assumptions to the question. If it's not stated, don't assume it's there. For molecules made up of the same number of monomers. So I have a protein with, for example, 4 monomers. That's an example. Okay, and a ribozyme, which is made up of RNA bases of also 4 molecules. The number of possible types of enzyme is greater than ribozyme. Now, this is true. Why? Because there are 20 amino acids. So for each one of these, there are 20 combinations. So that's 20. So that's 20 to the power of 4. Here, there are only 4 bases of RNA: adenine, cytosine, uracil, and guanine. So there are only 4 to the 4 possible combinations. 20 to the 4 is greater than 4 to the 4. So this is true. There are more possible enzymes than ribozymes. And so the answer is 2 and 3 only, which is D. Moving on. Some students checked graphs to show the Vmax and the Km. Which graph is correct? Km is substrate concentration. So here we have Km as rate of reaction. Wrong. Km is rate of reaction. Wrong. Km is enzyme concentration. Wrong. Km is substrate concentration. Correct. See. And that's just from a basic understanding of what Km is. The Michaelis-Menten constant is the substrate concentration at which the enzyme is at half of its maximum, at which the enzyme is at half of its maximum activity. And obviously Vmax is rate of reaction, but that's constant throughout the mole. So it is literally the definition of Km that helped me know this question. I barely even looked at the options. Moving on. Enzymes can be immobilized in alginate beads. Our A2 friends or our composite friends can help tell us how this is done in specifics. P5, I hope you guys have studied it, the A2 and the composite people. Anyway, what are the advantages of immobilizing enzymes than free enzymes? Common question. Less likely to denature when heated, correct. Why? Because the immobilization stabilizes the structure of enzymes. It is kind of like chaining. It's kind of like stapling a piece of paper in 4 corners to a larger piece of paper. It's more difficult for this piece of paper to change shape. Now, obviously, if you somehow manage to break the larger piece of paper, the smaller piece of paper will change shape. But it is harder. It is less likely to denature. So this is correct. We can eliminate speed. It is easier to reuse the enzymes, correct. Okay, it is easier to reuse the enzymes when making new batches. Why? Because I can put the enzymes in a test tube and I can pass the substrate through it, substrate solution through it, all the way down to make, and the enzymes are still there in the immobilized beads. But if they are free, then they'll be in the mixture and it'll be difficult to remove them and then reuse them. Then they're easier to purify. Same reason is here. Again, I can, even if I didn't use all the measuring cylinder stuff, I can filter them much easily and extract them from the product and just take the product and set up and keep the enzymes and reuse them. So all 3 statements are correct. So the answer is A. Now the diagram represents the arrangement of some of the molecules on cell surface membrane. P and Q identify two regions of one of the molecules, and R and S identify two regions of another. This is a glycoprotein. Protein because it has a large 3D definite shape and carbohydrate chains on it. So this is a glycoprotein. And this is a normal phospholipid membrane, which always contain oxygen atoms and always contain phosphorus atoms. First things first, the phosphate head we know from IGCSE chemistry. Anything that ends in 8 has oxygen. So P is immediately, P is there in all of them. What's the difference? The difference is whether Q is there or not. So again, one of the key parts. One of the key parts of identifying the answer is identifying the different elements in each question. So when looking at what contains oxygen atoms, I'm just going to look at exclusively at Q. Now Q is the fatty acid chain. Fatty acid chain, the residue, the remaining part, especially the part that's not near to the phosphate head, that is not near to the phosphate head, is just a hydrocarbon chain. It's just C bond H bond C bond A is C bond H, C bond H, C bond H. Whether it's a double, whether there is C double bond C or not, it is still just a hydrocarbon chain. Hydrocarbon, again, also in IGCSE chemistry, a hydrocarbon is an organic compound that contains hydrogen and carbon only. So Q does not. Now, so the answer is either C or D. Always contains phosphorus atoms. Now the difference here is whether P and S or P only. So let's look at S. S is a carbohydrate chain. Carbohydrates, carbohydrates, carbo, carbon, hydro, hydrogen, 8, oxygen, same reason as phosphates. So they have carbon, hydrogen, and oxygen. End of discussion. So they do not contain phosphorus. So the answer is P only. The answer is therefore C. Moving on. Adrenaline is a hormone that prepares the body for rapid responses by increasing the blood flow and increasing blood sugar concentrations. It is secreted from adrenal glands and acts on nearly all body tissues by binding to specific adrenergic receptors on the surface of the cells. Different tissues respond in different ways. With an example, nobody cares. What is the different responses of what could explain the different responses of the muscle to the adrenaline? I'm going to read B, C, and D first, and then I'm going to say why A is the correct answer. B, the adrenergic receptors on smooth muscles in the airways bind to different ligands. Now, ligand is just the hormone substance that it is the adrenaline. It is the cell signaling molecule. Okay. So obviously B is wrong. They bind to the same ligand, which is adrenaline. The question states it clearly. C, concentration reaching total distance of the adrenal glands and whether smooth muscles relax or contract. Now the adrenal glands, now contraction of smooth muscles and arterioles. Now these arterioles are even further than the ones in the lungs. They could be in the brain. Okay, they could be near your eyes. So the distance has nothing to do with it. Okay, your adrenal glands are over your kidneys. So which is closer? And even if we say it's further, and even if we say it's further or closer, there are also arterioles that are closer to the kidneys, to the adrenal glands, such as the ones in the kidneys. So this is simply nonsensical. Now, another thing that's important to note is that the concentration affects the severity of the action. So they could contract more or less. In that case, I could say maybe maybe C is to be considered. But the fact that, completely opposite responses, contraction in one area and relaxation in other area, no, that has to be some type of change within the cell signaling pathway itself. So this is also wrong on multiple counts. The binding sites have different tertiary structures. Now this is the same sentence as B. The binding sites of different tertiary structures means the binding sites have different 3D shapes. We know that tertiary structure means 3D shape. And if the binding sites have different 3D shapes, that means they bind to different ligands, to different substrates. different ligands, sorry, and we know that it's the same ligand. That does not mean that the receptors cannot be different. Here it says the binding sites. Okay, so that means one's like this, and another is like this. Now, this is adrenaline. For example, if they have different structures, the same adrenaline molecule will not fit into both different binding sites. So again, D is wrong. Now let's go to the correct answer, A. Smooth muscles have different types of receptors which initiate different responses. This is correct. When we say different types of receptors, that does not necessarily mean that the binding site is the same. This, let's just say this is receptor 1, and let's just say this is receptor 2, whatever. Both have the same shape here, which is a square or rectangle or whatever, okay? But they are different in their form of intracellular, in the forms of signal transduction, converting from extracellular signal to intracellular signal. Perhaps they have different G proteins attached or different, where they activate different secondary signaling molecules. This is correct. Again, when there are opposite changes or completely different changes in response to the same receptor, that means that there is a change in the cell signaling pathway. And that could just mean that certain pathways, certain enzymes within the cell signaling pathway themselves are different, or that the receptors themselves are different towards the intracellular site, towards inside the cell. So they cause different signal transductions. Signal transduction is converting from an extracellular signal to an intracellular signal, from a signal that's outside the cell to a signal that's inside the cell. We are running very late. What is the effect of red blood cell being placed into pure water? Here's the cell and here's the water. Now, pure water has very high water potential and the red blood cell has low water potential. So we move in by osmosis from high water potential to low water potential. So, and again, this is net. So not all the water enters and not all the water leaves. Most of the water enters and some leaves. A lot enters and some leaves. Someone reads, the first option says, less water leaves the cell than enters it. Less water. Less water leaves, so this is small, and the one that entering it is large, so this is correct. So the cell swells and bursts, correct. More water enters the cell than leaves it. Someone goes, oh yes, that's true. Does not read, so the cell becomes turgid. First off, when referring to turgor pressure or turgidity, we only refer to cells with a cell wall, and generally plant cells. This is not a plant cell, so the term turgid is wrong. Another thing to note is that the red blood cell does not have cell walls to withstand the high blood pressure. Does not have a sufficient cell wall or a sufficient boundary to withstand the high blood pressure and to become turgid. It's just going to burst. There is no period of time in which it's turgid. So B is wrong. Water can enter the cell, but is unable to leave it. Wrong. We said it's net. Water enters and leaves at the same rate, so the cell does not change. Obviously, that's wrong. So the answer is A. A scientist used a sharp knife to cut a cube from a potato. It had this mass. Scientists placed this cube in the sucrose solution and measured the mass. Sucrose solution, so low water potential in the potato. Oh, never mind. There's still more to the question. After 120 minutes, scientists measured turgor pressure of the potato cells, again, plant cells, the water potential of the potato cells and the water potential of the sucrose solution. Turgor pressure is the hydrostatic pressure within a cell that pushes the cell surface membrane against the cell wall. Again, cell wall presence means turgidity, so we can't use it on cells. Which value of potato cell turgor pressure, potato cell wall potential, and sucrose solution potential after 120 minutes are consistent with the results? Now we see here that the mass is increasing. And at the end of the experiment, the mass is constant. That means there is no longer a net movement of water to or from the potato cell. That means we have reached equal water potential. Okay, so equal water potential between the potato and the sucrose solution, the answer is B. immediately without even thinking. Now the trigger pressure is obviously going to be, this one doesn't really matter, it's just a thing. Now notice here, it's after 120 minutes that the scientists measure them. Beforehand, the potato cell water potential would have been lower than the sucrose solution water potential. So the answer would have been C, but this is after the measurement. Moving on. Keratinocyte stem cells are found in the skin where they are important for the repair of damaged skin. Which row shows how keratinocyte stem cells respond when skin is damaged? Very, very common error for people to assume that that stem cells repair damaged cells. No, they repair tissues. They repair tissues by replacing damaged cells. So they do not repair damaged cells, but they do divide by mitosis, obviously. So the answer is B. Four days after fertilization, the human embryo was found to contain 32 cells. How many cells will be there in the human embryo after completion of 4 more metallic cell cycles? Paper 1 is the only paper in which the examiner can misdirect you. 4 days after fertilization is absolutely useless. It just says 4 more mitotic cell cycles. Now we know that in each mitotic cell cycle doubles. The number of cells doubles. So 32. Let's do it one by one. 32 becomes 64. Becomes 128. That's the first division. That's the second division. Becomes 256. That's the third division. Becomes 512. That's the fourth division. And the answer is B. The photomicrograph shows cells in different stages of the mitotic cell cycle. Cell T shows metaphase. Which statements are correct? Cell T shows metaphase. Correct. We see all the chromosomes aligned at the equator of the cell, so cell T shows metaphase is correct. Anything without option 1 is wrong. D is wrong. DNA replication is occurring in cell R. This is incorrect. If we look at the surrounding cells, we see this is interphase because the nucleus, because the genetic material is still compact. R is prophase. There are clear strands. I can easily distinguish this strand and this strand and so on. So there are clear strands indicating that it is in prophase, and we know DNA replication occurs in S phase, which is an interphase of mitotic cell cycle. So 2 is wrong. So anything with 2 is wrong, and our answer is C. Congratulations, you finished the question. Let's read the other statements just in case. The amount of DNA in cell P is the same as the amount of DNA in cell T. The only time the amount of DNA changes is from before and after S phase. Now, since P is in anaphase, notice the clear V pattern of the chromosomes, and cell T is in metaphase. Both after S phase, they have the same amount of DNA. So again, 3 is right, and our answer is still C. The correct order of stages is SRTPQ. S, interphase. R, prophase. T, metaphase. P, anaphase. Again, notice the clear V appearance. And Q is telophase. I can no longer distinguish individual strands of chromosomes. Rather, I see a huge blob, either 2 huge blobs, and therefore it is the nucleolus forming and the chromosomes uncondensing and uncoiling back into chromatin. So this is correct. Which statement about nucleotides is correct? Adenine, guanine, or pyrimidines? No, they're purines. ATP is a phosphorylated nucleotide. A nucleotide has 3 components. The pentose sugar, whether deoxyribose or ribose, the nitrogenous base, and 1 phosphate group. To be phosphorylated means you have extra phosphate groups, and ATP, adenosine triose, tri meaning 3, has 3 phosphates. Purines have a single ring structure? No. Short name, long structure. Large structure, double ring. The uracil replaces thymine, not cytosine. The answer is B. The scientist analyzed the base composition of several species, which statements can be concluded using only the data in the table, and this emphasizes the idea. If not stated, we do not assume. Cytosine binds to guanine using 3 hydrogen bonds. Nothing here states any information about the number of hydrogen bonds. So 1 is wrong and anything with 1 is wrong. Okay, and the difference between C and D is 2. So I'm just going to look at statement 2 for the sake of time. Approximately 50% of the bases in any species are either adenine or thymine. Adenine 24 plus 23, that would be 47. 26 and 27, that would be 53. 29 and 29, that would be 58. 32 and 32, 64. 64, that's exceptionally high. That's greater than 10%, greater than our margin of error, even in physics. Okay, so I can't say approximately 50% in any of the species. The base composition varies between different species, that's true. Here I have 26%, here I have 17%. Here I have as low as 23% and as high as 32%. So it does vary. The number of cytosine bases in any of the species is approximately the same as the number of guanine. Of course, 25 and 26, 22 and 23, 20 and 20, 17, 17, 17, 17, 21 and 20, always within 1% of each other. So question 4 is correct. 3 and 4 only. The answer is D. 27. Gene from eukaryotes exert the DNA of the bacteria. It is not able to synthesize the polypeptide coded for by the eukaryotic gene. So we inserted into a bacterium some eukaryotic DNA. Now, why is it the case that the eukaryotic DNA is then inserted into the bacterium? The mRNA produced by the bacterium is different from the one produced by the eukaryote. This is true. When we say mRNA, we are referring to the mature RNA. So not the primary transcript. And bacteria cannot splice. They do not have spliceosome. There is a bit of an out of syllabus asterisk there. I'm not going to discuss it. So bacteria cannot splice. Only eukaryotes have the ability to splice. So the mature RNA produced by the bacteria still has the introns, while the mature mRNA produced by the eukaryotes does not have the introns. So therefore, 1 is correct. Eukaryotic genes contain non-coding sequences, the introns, that are absent from bacterial genes. This is true. Bacterial genes, splicing is when you remove the non-coding sequences from a gene and rejoin the coding sequences. We call the non-coding sequences introns and the coding sequences exons. There are differences in the genetic code between bacteria and eukaryotes. This is obviously wrong. Why? We know 3 statements about DNA, and they're in my notes. Let me see if I have them. Nope. I closed my notes. Anyway, 3 statements. Degenerate, universal, does not overlap. 2 is the key point here. Genetic code is universal across all species. So 3 is definitely wrong. So anything with 3 is immediately wrong. 1, 2, and 3 is wrong. 1 and 2 is wrong. 1 and 3 only is wrong. So we're left with B and D. We know 1 is correct. So the answer is B. I'm going to speed up a bit because we're quite late. Why do some gene mutations result in the same synthesis of the same polypeptides? Degenerate genetic code. That means multiple triplets of bases can code for the same amino acid. Ribosomes correct mutations. No, they don't. The mutated bases will not be transcribed. What's stopping them from being transcribed? Non-overlapping means that the same triplet of codon, the same triplet of bases did not code for one amino acid here and a different amino acid. Each triplet of bases There are no triplets of bases that code for two amino acids. No, one triplet, one amino acid, one amino acid, multiple triplets, but one triplet, one amino acid. Mutated exons will not be transcribed, nothing stopping them. Mutated exon will be removed. Exons are not removed. Introns are removed. This is also wrong. Most amino acids have more than one DNA triplet code. That's correct. That means that if there is such a subtle mutation, which we call a silent mutation, there is no change. What determines the rate of water movement from the roots to the leaves of a plant? This is by transpiration pull, so anything that increases the rate of transpiration will also increase the rate of movement. Do they apoplast? No, doesn't matter. Less negative water potential in the leaves. Less negative means a higher water potential. So why would water move from the roots to the leaves? Rather the opposite. Less negative means higher. We know that water moves from high to low, so it would move down, not up. So B is wrong. Changes in the specific heat capacity of water, that has nothing to do with it, and the evaporation of water from the mesophyll cell walls, that's the definition of transpiration. If transpiration increases, transpiration pull increases, and water movement increases. I answered this non-overlapping means that the same triplet of bases does not code for more than one amino acid. Moving on. Transfer sections through three different types. Someone might want to add Sue or whatever, because this is an important question. The photomicrograph shows the transverse section through three different types of blood vessels. All are shown at approximately the same magnification, so they're the same size. What we're going to do is we're going to look. First off, type 3 has a very wide lumen. It's a vein immediately. And obviously, the wall has more than one layer. So it's not a capillary. I'm pretty sure that's clear from the wide lumen. So type 3 is vein. Now, type 1 and type 2, elastic or muscular. Now, elastic arteries, the elastic tissue, the elastic fibers will be clearly visible in the tunica media. Here, you can clearly see these black squiggly lines all around. They're very visible. Here, so type 2 is obviously an elastic artery, and hence type 1 is a muscular artery. The answer is B. The diagram shows transverse sections of parts of a plant. They are labeled 1 to 6, which row shows two tissues that mainly transport water and two tissues that mainly transport sucrose. Mainly transports water is the xylem. Now in leaf cells, xylem is up and phloem is down. Here's in everywhere else, xylem is the cells that are closer to the middle. So 4 is xylem, 3 is phloem, and 5 is phloem, 6 is xylem. Mainly transports water. 1 is xylem, yes, and 3 is phloem, no. 2 is phloem, no. 3 is phloem, no. 4 and 6, yes. 4 is xylem and 6 is xylem. Mainly transports sucrose. 2 and 3, both are phloem. Moving on. Mass flow and phloem. T is the phloem. Concentrated sucrose solution means low water potential, and dilute, that's higher water potential, and this has the highest water potential. Water moves in or out of X or Y as a result of osmosis, or as a result of hydrostatic pressure. Sucrose solution then moves through the tube that joins X or Y. Water moves into X to increase hydrostatic pressure as a result of osmosis. So X has higher hydrostatic pressure, which forces the solution to go from X to Y, from concentrated to dilute. And it already gave you a hint. Demonstrated mass flow in the flow. So it goes from where there's a lot of sucrose to where there's few. So it goes from X to Y, and the water potential in X is lower, so more negative. Which statement about the transport of sucrose in plants is correct? The proton gradient is maintained by active transport, correct. Sucrose moves through plasmodesma by passive diffusion from the mesophyll, no. To the companion, no. It moves from the companion to the phloem sieve tube, not from the mesophyll to the phloem. 30 is B. Again, notice here, very visible elastic tissue. See the black, very clear black fibers? These are the elastic tissue. I'm doing it right now. Okay, type 2, very clear black fibers. These are the elastic fibers in the tunica media, and these are, when they're very, very clear, they are elastic arteries. Here, and you can barely see one or two. Obviously, this one has way more. What you can see more of here is the gray or pink background, the muscle, or so to speak. So the type 1 is muscular, type 2 is elastic, and type 3 is vein. Again, here, as we were saying, it moves from mesophyll to companion cells by secondary active transport. It moves from the companion to the phloem seed tube through the plasmatosomatic acid diffusion. ATP synthesized from the mitochondria in companion cells provide energy for the active transport of sucrose. No, by secondary active transport provides active transport for the movement of protons. Water enters companion cells by active transport? Absolutely no. This is just nothing here is correct. It's not even familiar. When you see something that's not familiar, always know it's probably going to be wrong. 34, blood smear, which identifies the labeled cells. This is a blood smear, so we have monocytes, not macrophages. Kidney-shaped nucleus, number 1 is monocyte. Number 2, lobed nucleus, neutrophil. And number 3, very large nucleus taking up most of the cell space, that's lymphocyte. The answer is A. Which draw identifies properties of water important for its role in the transport? High specific heat capacity. It's true. Water has that, but it's not important for transport. So this is X. Latent heat of vaporization. Water has that, but it's not important for transport. Also another X. But it is important for the glucose and the ions to be able to dissolve in the water. Our answer is D. Question 36. Which substances will increase in concentration in the plasma and the capillary surrounding muscle tissue? Hemoglobin is in the red blood cells. So one is wrong, and our answer is D. Moving on. Hydrogen carbonate atoms. Now, carbon and hemoglobin will increase. Now, closer to muscle cells, more carbon dioxide is produced, more carbonic acid, more hydrogen carbonate ions. So the answer is 2. Carbaminohemoglobin is in the red blood cells. Here it's asking about in the plasma. They mean directly in the plasma. They do not mean inside the red blood cells. Because I mean, in that sense, if I have a lunchbox in a boat and the boat's in the ocean, is the lunchbox in the ocean? It doesn't. If I say, I want stuff that's in the ocean, you have a lunchbox on a boat. Are you going to say, oh, the lunchbox, because it's on the boat in the ocean? No. Please. Which statement correctly describes the structure of the human gas exchange system? The wall of an alveolus is only The wall of the alveolus is only 1 cell thick, correct, and contains smooth muscle, absolutely. 1 cell thick to shorten distance for faster diffusion. Smooth muscle, no. The alveolus does not have smooth muscle. It rather has epithelial cells, squamous epithelial cells. The wall of the bronchiole consists of cartilage. No, only bronchi and the trachea have cartilage. The wall of the bronchus has a folded inner layer of ciliated epithelium which may contain goblet cells. True. The wall of the trachea contains many ciliated epithelial cells and is supported with complete rings of cartilage. No, there are no complete rings of cartilage. At max, we have in the trachea C-shaped cartilage, and the space there is for the small space is for the esophagus during peristalsis, so the food doesn't get stuck. So the answer is C. Three statements about the pathogen that causes TB are listed. It inhibits some immune responses, such as phagocytosis. It reproduces slowly and is able to become dormant within infected tissues. Why does treatment take a long time? 3 is correct. Because the pathogen can just sleep inside the infected tissues and there are no symptoms. And so we are not sure whether or not the pathogen is gone. So we continue treating to be sure that it dies. The pathogen reproduces slowly, also correct. Because if the pathogen reproduces slowly, same reason as 3, we are not sure if 3, we are not sure if the pathogen is completely dead or not. And also, because of its slow reproduction rate, it can slow itself down and then it just spreads really slowly. And so the antibiotics cannot disrupt, and so the antibiotics are not able to ensure full treatment, except after a very long time, to ensure not one single pathogen remains, and that just spreads very, very, very slowly. Pathogen can inhibit some immune responses. That's not relevant at all. Why? Antibiotics work directly on the pathogen itself. If we go back to our antibiotic chapter, we know the penicillin breaks the cell wall. What does phagocytosis have to do with antibiotics? It doesn't. If it had antibodies, maybe, but this is antibiotics. So 1 is wrong. So the answer is 2 and 3 only. 38D. Last 2 questions. During an outbreak, immediate protection is needed. What type of immunity could provide immediate protection? It will take a very long time to work. We know this. So A and B are immediately eliminated. Natural immunity does not provide. Now, natural passive immunity, the issue with it is that it cannot be administered immediately. Natural passive immunity is either through the placenta or through the colostrum of breast milk. It is not something active. Artificial passive immunity is giving antibiotics. It's giving antivenom. It's giving monoclonal antibodies. Now, which statement describes the responses of T lymphocytes? It did not specify whether a T helper or a T killer. It will specify in the options. Body cells are infected by viruses. T helper cells destroy the infected body cells by phagocytosis. T helper cells destroy infected cells by phagocytosis. Not a single statement in the sentence. T killer cells are the ones that destroy the infected body cells, and they do it by secreting perforins. T killer cells bind to antigens on the surface of infected body cells. T helper cells release cytotoxins. Helper release cytotoxins? No. T killer cells are also called T cytotoxic cells because they release cytotoxins, which kill the cell. T helper do not. T killer cells secrete antibodies? No, that's plasma cells. So our answer is B, and congratulations, we're done. Where's the whiteboard? Okay. Now I'll answer some of the questions that you guys sent in the thread. Let's open it. And let's go from the top. Let me forward this to. Okay, I'll stop sharing. Okay, here. Why is A wrong? A is wrong because it says that the observation that test tube stays clear. Now, if the hydrochloric acid will denature the proteins and cause the membrane, both the outer membrane, the cell surface membrane, and the tonoplast, I turned off screen share. Open the chat now. Okay, and the tonoplast, then the tonoplasts also have membrane proteins. So the anthocyanin pigments or the pigments or whatever will diffuse through the tonoplasts regardless, and they will exit. So the answer, so A is wrong because it says that the test tube stays clear. It doesn't. The treatment and the explanation are correct, but the observation is not. Next question is this one. Which part of the graph represents the start of atrial systole? Atrial systole means that the atria are contracting and the ventricles are filling up. Okay, the ventricle volume is decreasing now. The contraction of the ventricle, that's ventricular diastole. Immediately after ventricular diastole, there is just that's A to B is ventricular systole. The ventricles are contracting. B to C, we know that the stage immediately after ventricular systole is diastole. Both the atria and the ventricles are relaxed. That's why it very slowly, the ventricular volume very slowly fills up until the atria contract and they force just a bit more. So C is the start of atrial systole. You could do this by processes. A to B, we know that the ventricular volume is decreasing. That's going to be ventricular systole. Then B to C, diastole. Then C to D, atrial systole. And then it repeats D to A, like A continues, D to A would be ventricular systole. You could go in order if you're not sure, but there's also like a reasonable explanation for it. Now to the next question. No other questions. Okay. Cardiac. Acetylcholine's question. Is the word maintain and withstand specific for any one type of artery? Or is it a common word to describe both artery sections? Neither. They are different words. Like maintain means to ensure that the blood pressure remains the same. Okay, it's why there's narrow lumen, for example. It ensures the blood does not decrease, to the ability to not burst under high pressure. I can, and it is referred to the thicker walls. Make sure that the how about now? No. Okay, I'll explain anyway. Maintain just means keeps the blood pressure as it is. Withstand means to keep the means to be able to not burst under high pressure. They describe different actions, but they do not describe different types of arteries. I can use them for the muscular and the elastic arteries. We use maintain when referring to the narrow lumen. We use withstand when we're referring to the thick walls of the arteries, regardless of what type of artery. I hope that's clear now. Next question. Surface area calculations. The key thing about surface area calculations in paper 1 is that you work by the shortest distance. If it says the time to reach the center, you find the cube or whatever with the shortest distance to the center, because then most of the liquid will go through that, will reach it faster. And as for the cardiac cycle graphs, here I'll share my screen now again to describe them. Okay, here is the cardiac cycle and the graph word. Can you guys clear the screen? Hello? Okay, perfect. So the first thing that happens is atrial systole. The atrial muscles contract, this increases the pressure. We have this very narrow increase. Again, yellow is the atrium. Now the blue also increases because there is blood moving to it, so the pressure is increasing. Okay, but it is the atrium still has a higher pressure. The moment the ventricles have a higher pressure, they contract. So the atrial muscle relaxes. And this is stage 1, sorry. This is 1. Okay. The atrial muscle relaxes. Okay. And due to now blood flowing to the atria and the rebound of the ventricles being closed, that's why there's this very subtle increase. Now, obviously, it goes down again. But the ventricles keep increasing until they're above the pressure of the aorta or the pulmonary artery. Right here. In this case, the semilunar valve opens, and again, the ventricles are still contracting, and are, at this point they're contracting, and then they go down, they're emptying now. Left or right, you look at it. The thicker let's see a larger diagram of the heart. Thicker wall for the ventricle is the left. Thinner wall is the right. And generally, it's going to be in this orientation. Thicker is this side, and thinner is like left is this side, and right is this side. But generally, you can determine it by from the graph. It's given here, like it says left atrium and left ventricle. The question usually specifies. For the right, the intensity is just lower, but here we're talking about the left area, like the left side of the heart, just to simplify stuff, so we don't have to focus on the right side as well. Keeps everything nice and simple. Now here, the semilunar valve closes, and we're in the period of diastole. Blood flows into the atrium, which is why the atrium is increasing, and blood flows into the ventricle, which is why the ventricle is very slightly increasing. This is the cardiac cycle. Okay, and here it is in like a large diagram form if you want to take a screenshot of this. Okay, are we done? Any other questions? Let me check. Yes, it has. You can watch the recording afterwards. All right. Thank you all. And goodbye. Good luck on paper 1. I wish you all the best of luck.